Steve, what's the first data type given?
That'd be nominal.
It wouldn't be nominal. Maddie, why not?
It would be ordinal because you're expressing things in like a living system, times better.
Okay, so they're categorical data, but they can be ranked from lowest to highest. And so they go beyond nominal to ordinal because they can be ordered. Brady, what about the second one? Pounds of corn in a bushel of corn.
I put a ratio
Why is that?
because, I mean, like, taking me compared like 10 pounds is twice as heavy as 5 pounds.
Exactly, so you can divide it, and so that's the test of ratio data. The whole reason that they're called ratio data is because you can do that particular mathematical operation on them. Colors, Chase.
Nominal, because it's categorical and
It's-
they don't relate to each other.
It's categorical and you can't order them in any particular way, in any objective sort of way. Good. Connor, what percentage of your day do you feel anxiety?
It's either ratio or ordinal. Anytime ratio.
Okay, explain what you mean by that.
Well, it can be subjective on a personal recording, so I think.
So it is subjective because you're asking somebody their opinion about how they feel.
Yeah.
But in terms of the way they express the data,
Ratio.
why is it ratio then?
Because if somebody experiences 20%, 40%, 40% is twice as much as 20%.
So a person who experiences 40% of their day feeling anxious is twice as anxious as somebody who is experiencing anxiety only 20% of their day. And so even though it's subjective and even though it's trying to quantify this thing that may be difficult to quantify, the fact that you're asking the question in a quantitative sort of way like that basically says, well, I don't know what makes you anxious, but if you're spending 40% of your day feeling anxious and another person is feeling anxious only 20% of the day, then that's a twofold difference in anxiety. However, each of those individual people is expressing it. Now, how would we ask the question if our goal was to get ordinal data?
Same as the first one on a scale.
Yeah, you would ask them to express what their level of anxiety is on like a 5-point scale from not anxious at all to extremely anxious or something like that. I'm just blanking on Mason Brooks today. Had you in an entire class before and I can't grasp your name out of thin air for some reason. Number of eggs produced by individual chickens in your backyard chicken flock.
That would be a ratio since you have meaningful multiples, right?
Yeah, so you can have one chicken that was producing 5 eggs per day, another chicken that was producing 2 eggs per day. Those can be divided, and you can figure out how much more one chicken is producing than the other. Karen, number of seconds required to run 100 meters.
That's the ratio data.
Yeah, that's ratio data because somebody running the 100 in 9 seconds is much faster. It's half or twice as fast as somebody who runs it in 18 seconds. And so body temperature in lizards, Bryce.
Interval,
Interval, why?
because it's like can be mathematically compared, but it's the heat is relative.
Okay, so it's on this kind of arbitrary scale, and doubling the value doesn't necessarily mean that you have twice as much of whatever the thing is that's being measured. Strangely enough, when I was putting together this list of examples, the one that is hardest for me to find examples for is interval. Because oftentimes numerical data, quantitative data, once you cross the line from qualitative data to quantitative data, a lot of the quantitative data that we come into contact with actually are ratio scale data as well as interval scale data. So finding quantitative data that are really only interval scale data was actually something of a challenge for me, and so I fell back on the old standard, which is temperature. Good. So just to remind you where we are and where we're headed towards a semester of doing statistics. before we can get to that, we have to understand the fundamental relationships between populations, the samples that we draw from those populations, and ultimately the inferences that we can make based on those samples. And so if we have a population here, we know from the central limit theorem that if we go and sample repeatedly from this population over and over and over again, for each of those samples, we calculate the sample mean, we save all those sample means, and then put them into a distribution. We know that a distribution of sample means will be normal. And this will work, assuming that we have sufficiently large sample sizes in our samples for any shaped distribution. I could draw distributions of all kinds of wacky shapes all day long, and assuming that our sample size is large, then the distribution of sample means will be normal drawn to this population, regardless of what the shape of that original population distribution is. Now, n equals 30, sample size equals 30. This is kind of a magical number. One of my graduate level statistics professors told me this. This is a guy named Patrick Phillips, who now teaches at University of Oregon. In UT Arlington when I was there. He said 30 is closer to infinity than it is to 0, because in terms of being able to produce this distribution of sample means that is normal from any wacky shaped distribution, once you hit 30, you're kind of there. Adding 10 more samples to your sample size doesn't really gain you any ability to do this. Adding 20 more samples doesn't gain you much more ability to do this. And so your ability to sample from this and have it give you a distribution of sample means that is normally distributed kind of plateaus out at about 30. And so gathering samples larger than 30 doesn't actually gain you much statistical power on top.
So 30, is 30 the number of observations? I got 3 things rolling around. Is 30 the number of observations in the population, or the number of observations in each sample, or the number of samples?
Number of observations in a sample.
Okay.
The population can be very, very large. Our whole point of sampling from the population is that the population is too large to sample the entire population as a whole. And so, if the population was only composed of 30 individuals, why would you sample that population? You would just measure all 30 individuals, and then you wouldn't have a sample, you would actually have measured the population. Yeah, it becomes a census at that point. There's no point in calculating confidence intervals, et cetera, because you've measured the entire population. You don't need to estimate what the mean is. You know what the mean is. It's the population mean at this point, because there are only 30 members of your population, and you've measured all of them. You know what the answer is. The average height of that population is whatever the mean of the population is. Now, if the population were 30 and you only sampled 15, then once again, that sample mean is an estimate of the population mean. But really and truly, if the population is only 30 individuals, you would just go and measure all 30 individuals, assuming that you can find them. This is actually an important distinction. that I had to make to a dean of this college once, because he made the mistake of asking me to analyze some data, and their data that you're going to analyze is your final project in here. There was a time back in the early 2000s when there was a question as to whether or not we were paying faculty at Williamsville College equitably. And since he knew that I had some skills with statistics, he asked me to analyze the data. And that was probably a mistake of his, because at that point, what he wanted to know in particular was, he had a set of questions. He wanted to know if the pay that we were paying assistant professors, associate professors, and full professors were kind of comparable in terms of the gaps, because full-time, full professor faculty were complaining about what they called salary compression, because once you get to full professor, there's not another promotion event. And so you get promoted to full professor, and then your opportunities for getting a raise, because you go to fuller professor, there's no such thing as fuller professor. And increases in salary in academia are tied to promotion events. So once you get to full professor, you kind of peak out, and then you're just kind of stuck there. And when you hire new faculty in, oftentimes you offer them salaries that are tracking the salaries that are being offered at other places. So as entering salaries are rising, there's no mechanism to make full professor salaries also rise. And so what happens is assistant professors come in, they get raises added on to their already raised salaries because they were hired more recently. And so assistant professors' salaries creep closer and closer to full professor salaries, and so the whole salary scale gets kind of compressed. So that was one of his questions, was what's happening to those gaps between assistant, associate, and full professors. Another fundamental question that he had was, are female faculty being compensated comparably to male faculty, or is there a gender gap at Williamsville College? And then kind of the third question that he wanted to know is he wanted to have some idea as to what differences among different departments were in terms of compensation. Some departments have to pay people more to come and teach because they could be, for example, out in the business world making scads of money, and they choose to come and teach instead. And so I was tasked with answering those three questions. And so what I got from him was I got a data set that basically had everybody's salary at the college, and it was all divided up by department. And so in an effort to keep the data anonymous, it was stripped of all identifying information, except that it also had the department of the person. So had I wanted to, it would have been pretty easy to go and figure out who was making what, but in reality, I didn't really care as long as I could divide people up by department so I could answer one of his questions. Well, the problem with him asking me to do this is that he wasn't asking me to use Williamsville College data to speak to a broader population. He was asking me to use William Jewell data to speak to William Jewell. So he delivered to me the entire population that he was interested in. And so when I calculated the average salary for assistant professors and the average salary for associate professors and the average salary for full professors, those weren't estimates of the average salary of those three groups. Those were actually the true population means for those groups, because he had delivered to me the population data. So if assistant professors who were male were making $36,856 per year, and the females were making $36,854 per year, that's a difference. It's a real difference, because there's no error associated with those two things. Males are making, on average, $2 more per year than females. And that's different, not because I calculated a confidence interval around those means, and the confidence interval was so narrow that I can tell the difference between 36,856 and 36,854. They're different because I have the entire population of male assistant professors and I have the entire population of female assistant professors. And if those two numbers are different, then those two populations are different. Now, it was more like a $2,500 difference. And I concluded, we're not paying women as much as we pay men, by about $2,500. And because you gave me population data, that's a real difference. Not only that, that's like a down payment on a car kind of difference. And he got up in front of the faculty when he presented these data to the faculty and tried, and so I plotted 95% confidence intervals because it's just a habit that I've gotten into. And he tried to parlay this into presenting it as if these differences weren't really real because of the 95% confidence intervals were fairly wide, because we had a couple of faculty in the math department who were stuck at the assistant professor level because they only had a master's degree, but they had been here for like 30 years, so they occasionally got cost of living adjustments, so they were making a ton of money in terms of assistant professors relative to people like me who was an assistant professor at the time, who was not making very much money because of the academic level that I was hired at. And he tried to parlay it into that there was some uncertainty about these data, and maybe this difference wasn't actually real. Unfortunately for him, I was sitting in the audience at the faculty meeting when he presented this, and I raised my hand and I said, actually, what you just said isn't actually true. Because you gave me the entire data set, those are population means, which means that there is no error associated with them. That is what they are. And that $2,500 difference is an actual real difference. Now, the question isn't how much uncertainty is there around these numbers. There's no uncertainty around these numbers. The question is, do you feel that we should do something about these numbers? Because they aren't samples, it's a population. You gave me the population. Now, you can say we're going to do nothing about these numbers. That's perfectly fine. You can say we're going to do something about these numbers. That's perfectly fine as well. I put it to your peers on the faculty whether or not they would favor one of those decisions versus the other. Needless to say, the next year when I went up for tenure, it was less comfortable than I could have been. The difference between a population and a sample. If you have the population and the population is small enough, you can actually just forget statistics, sample the entire population, and the number that you get is the number that it is. Because you know everything there is to know about the population. Seldom do we in statistics have the opportunity to sample the entire population. When you can, do so. When you can't, invoke statistics. So, we have populations, we have the distribution of sample means. We generally don't know what this distribution of sample means is because we don't have the time and the money to go to the population, repeatedly sample it, and then create a distribution of sample means. Because if we had the ability to do that, if we had the time and the money to do that, we would actually just get the distribution of sample means. We know that the population mean is equivalent to the mean of the distribution of sample means. And once again, we've just taken a brute force sampling approach to estimate very precisely the population mean. And once again, we wouldn't do statistics on that. We would just do a lot of resampling of the original population until we had resampled it enough that we know what this distribution is, it looks like a normal curve, and we would know that this is the population mean, this is the population mean, the central limit theorem tells us that. And then we wouldn't, once again, invoke any statistics other than the central limit theorem. But once again, seldom do we have the time and money to do that repeated sampling of the population that we're interested in. Typically, we go and we sample the population once. Because that's what we have the time and the money to do. So now the question is, how do we relate this sample back to this distribution of sample means that we know relates back to the original population in some real way? Yeah.
So we were talking about the shape of the distribution.
I guess I am. I'm not gonna expand your question. I'm not sure what you're asking.
You're asking the disposition of data, right?
This is a sample of data, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. We have an N of 14, and we want to be able to take this sample of data and ultimately go back and say something about the population that it came from. But what we might want to do is not necessarily say something about the population that it came from, as much as we might want to just be able to say something about the distribution of sample means that we're drawing this population. So from this sample of size 14, we can calculate the average, the sample mean. An easier thing to make an inference about is how this sample mean compares to this distribution of sample means, rather than how this distribution mean relates back to the population mean. Does that make sense? Okay. Other questions?
See, you only took 14 in that sample, that's an issue, right? I mean, that's a.
Actually, 30 is closer to infinity, but so is 20 closer to infinity.
It'll do.
15 is closer to infinity than it is to 0 in a statistical sense. Once you get down below 10 is when I start to have questions about your ability to take this sample and then say, okay, if we sample repeatedly from this population at a particular level, n equals 5, then things get to be a little more sketchy. You can simulate this for yourself. You have the R script that I use for showing you how the central limit theorem works. You can run that R script on either a uniform or an exponential or a binomial or any other kind of distribution, and you can vary the population, you can vary the sample size from a sample size of 2, which is kind of the minimum that you need in order to calculate a variance, all the way up to whatever large sample size that you want. So you could set sample size at 2, set sample size at 5, set sample size at 10, set sample size at 15, set sample size at 20, set sample size at 25, 30, and you can then look at the distribution of sample means that comes from that, because the computer is repeatedly sampling at each of these sampling intensities. And you can kind of decide for yourself where the cutoff is for when you would start believing people's parametric statistics. The other thing that you can do is you can just go and get a t-table. So let's just go and get a t-table here really quickly. I don't generally carry these around with me because they're so easy to just find on the internet. So this is a t-table, and basically what it's giving you is a 2-tailed probability of 0.25 is essentially a 1-tailed probability of 0.5. So at this level, what we're basically saying is that we're interested in this distribution. We're going to put 2.5% of our observations out in this tail and 2.5% of our observations out which leaves 95% of our observations here in the middle. And so we'll look at this column here. Well, when you have 1 degree of freedom, which means you have 2 observations, the t-statistic, the statistic that you would multiply your standard error by to get the confidence interval for this thing is 12.71. But add 1 more individual to your sample, so go from a sample size of 2 to a sample size of 3, that drops to 4.3. Go from a sample size of 3 to a sample size of 4, it drops to 3.8, or 3.1. Go from 4 to 5, 2.76, 5 to 6, 2.57, 2.44. So there comes a point here where the multiplier that you multiply to create the confidence interval for this thing, we haven't talked about confidence intervals yet, there comes a point here at around 6 or 7 sample size where it ceases to change very much. To go from a sample size of 7 at 2.44 to infinity, the t-statistics goes from 2.447, is that right? Did I remember that right? Oh, yeah, it is right, to 1.96. This is a sample size of 7, this is a sample size of infinity. So it doesn't really change all that much once you get to a certain sample size. If we come down here to 30, it's 2.042. That's what Patrick Phillips meant when he said 30 is a lot closer to infinity than it is to 0. The t-statistic that you use to calculate the confidence interval doesn't really change very much. As you go from 30 to infinity, which is 1.96. So students oftentimes in undergraduate research will come and ask me, you know, is my sample size big enough? Well, I'm like, big enough for what? Big enough to tell differences among 2 samples? Well, it depends partly on how much variation there is in the samples that you're taking, and it also is going to be determined by how big the differences are that you're trying to detect. If you're trying to detect really big differences, you don't need huge sample sizes to detect that, because even those confidence intervals, even though they're going to be wide, they're small relative to the differences you're trying to detect. And so you can get a sense by running simulations in an R script that I give you at different sample sizes, or you can just go and look at a t-table and say, okay, as sample size increases, what happens to the t-statistic? Is it changing a lot now, or is it changing a little with each incremental addition of a sample? My animal behavior students last week read a paper. In that paper, the researchers used a thing called the Mann-Whitney U-test, and the students were like, why did they use the Mann-Whitney U-test? I'm like, well, they used a Mann-Whitney U-test because they had really small sample sizes. They had a sample size of 3. That's at the low end of things. And even though you could invoke the central limit theorem to say that if you take repeated samples from this population at a sample size of 3, it will give you an estimate of the distribution of sample means, that's getting to that low number of samples where your ability to make this argument begins to erode. And so rather than assuming that this distribution of sample means is normal, they use the non-parametric method that allows you to relax that particular assumption. The Mann-Whitney U-test is just the non-parametric equivalent of a t-test. And unfortunately, the 2 students that I was talking to in my office about this were not taking this class, and so it was a little more difficult to explain that to them than it would be to you guys now. You guys would see the Mann-Whitney U-test. You would say, what is this Mann-Whitney U-test that these guys are employing? I would say, oh, that's the non-parametric equivalent of a t-test. Why are they using it? They're using it because they don't want to have to assume that drawing from their population would give them a distribution of sample means that is normal. Because they have small sample size, and we'd be like, oh, okay. And then we would move on, rather than me kind of going and giving them a mini lecture on central limit theorem, which I didn't do very effectively as it turns out. So the t-table gives you some insight into this. You see how rapidly it falls off, and then you get to a point where it's just really not changing very much. At that point, adding more samples doesn't really gain you a lot. It gains you a little bit. But if it costs a lot more money to add 5 more samples, then you might just say, well, I don't need 5 more samples. This is going to be good enough for the task that I have at hand. Questions? When I ask you for questions at this stage, I really need you to tell me if you're not understanding what I'm going through, because this is the basis of everything else we're going to do in class this year. Let me illustrate to you why that's the case.
Big Paul.
Yeah.
So if you have a sample size of 2, then the difference would be 1, correct? Or is that?
Yeah, degrees of freedom would be 1, yes. It's always 1 minus your sample size, yeah. Yep.
Is the sample size given to you, or you have to like figure that out?
You'll know what it is by looking at the data. It's just the number of observations that you have. If you're doing work, at the beginning, you should try and figure out what sample size you need to have in order to detect the kind of size difference that you're trying to detect. And so there are things called power calculators. We haven't talked about alpha really, so we haven't talked about beta, but there are 2 things that you have to think about when you think about doing statistics. One is, what's the probability of type 1 error that you're willing to put up with? And then the other question is, you have to decide what level of difference you want to see, for example, between 2 means. What level of difference are you trying to detect? That level of difference will then determine how many samples you need to gather in order to detect that difference at a particular type 1 error rate. We're actually going to be talking about that on Monday. And so you can, if you have some idea of how variable your data are going to be, and you know the kind of difference that you're trying to detect, you can then figure out how many samples you need to detect that difference with that much variation. Seldom do people use power calculations in this way. Usually they use power calculations after they've done an experiment, found that they didn't get a significant result when they thought they were going to get a significant result, and then they ask, given my data, what's the smallest difference that I could have detected? And they find that that smallest difference that they could have detected is much larger than the difference that they actually observed. Well, of course, because if you could have detected that small difference, then you would have gotten a significant result. So doing the power analysis after the fact is kind of stupid, because it just tells you what you already know, which is that you didn't have enough power to detect the difference that you observed. So the smart thing to do is to do those calculations beforehand, so that you can say, I think the difference is going to be about this big of a magnitude of difference. What sample size do I need to have to detect that difference, given how much variability I expect to see in my data? And so one of the nice things about doing preliminary data, preliminary experiments, is it gives you some idea of how variable your data are, and in order to do these calculations about sample size, you need to have some estimate of variability. Because if your variability from one sample to another sample is really small, then you can detect really small differences with pretty small sample sizes. If your variance is considerably bigger, those differences get obscured by that variability, and so you need more samples in order to determine the difference of a particular size. Then you can say, okay, I need to do this many trials of an experiment in order to detect this magnitude of difference that I'm thinking is important to me. Usually, people don't do that. I was in Puerto Rico a number of years ago talking to my postdoctoral advisor. This was after I had left his lab and came to work here. And he shows up at the field station one day while I was there doing field work, and he's like, what time are you going to get back from the field today? And I said, I don't know, probably 3 o'clock. you some beers and talk some stats with you. I have a doctoral student whose committee I'm serving on, and we're having problems with her data. So I'm like, sure. So I go to the field, I get back, he's there, get showered and changed, we sit on a porch at the field station, and he kind of lays out what this person's experiment is, and we spend about 3 hours going through this student's data, looking for an appropriate way of analyzing it. And we finally come to the conclusion that he had already come to, and he just wanted a second opinion, which was this person had collected data in such a way that it was impossible by any method that we knew to analyze it. And at the end, I just turned to him and I said, you know, Jess, sometimes bad data happen to good people. And this is one of those cases. There's just nothing that either of us knew, and we had, between the two of us, a lot of experience in doing this. Neither of us could figure out a justifiable way of handling these data. The problem with that is that this student was in the process of writing the dissertation. Fieldwork was over with. She was done. The problem was that she didn't go and talk to a statistician before she designed her sampling, and a statistician would have looked at her sampling design and said, you can go and sample like that, but there's no way that you're going to be able to do anything with the data that come out of it. Far too often, even those of us who know things about things don't think about things like, what is the minimum sample size that I need in order to detect the difference that I'm looking for? We don't ask those questions as often as we should before we go and collect our data. We generally ask those questions after we've gotten the data and the data turned out to not be what we thought they would be. And sometimes that's catastrophically bad, sometimes it's just a minor irritation. One of the purposes of being in a class like this is that you avoid the catastrophically bad and you are just occasionally irritated. All right. I added this, so I updated the current PowerPoint, and I also added another PowerPoint to today's activities. I think it was today's activities. I am not a genomic scientist. I do not work with genomes typically, but I started doing some population genetics work a number of years ago, back in 2011, 2012. Oh, there's Patrick Phillips. This is my old professor. The guy who said 30 is closer to infinity than it is to 0. When people do genomic level studies, what they do, and you don't have to pay attention to the first part of this, you just have to pay attention to the end. What they do is they go and they sequence a bunch of genes, and then they take these genes and they line them up in a genome. And so this is a genome of a worm, and the colored bars represent different alleles of the same gene on paired chromosomes, one that they got from the mom and the other that they got from the dad. And I've always wondered how you test the significance of when a gene is represented more in a population than you would expect by just random distribution of genes in a genome. And so you generally do this by calculating a genetic statistic called FST. It's the degree to which a gene is represented in a subpopulation relative to the total population. That's what the S and the T are. So if you have a species that is distributed across 30 different populations, you can calculate what that gene frequency is, what that allele frequency is in population 1, what it is in population 2, what it is in population 3. That's going to vary from population to population because of the vagaries of the history of those populations. But then you compare that to what's the distribution of that allele in the total population. And for some genes, the FST, a lot of the variation is from subpopulation to subpopulation relative to the distribution in the total population, which means that there's a lot of variability in that allele. And then there are other places where there's not very much variability at all from subpopulation to subpopulation. And so as I was learning about these techniques, I was trying to figure out, so they have these statistical tests that they use to determine when there's more or less variation in a population than you would expect. And I couldn't wrap my head around it, until I got to this paper by Hohenlohe, Phillips, and Cresco. And then they showed me this figure right here. And when I looked at this figure, I was instantly like, oh, I see what they're doing. They're basically looking at the FST for each gene across the entire genome. They're compressing this whole thing, squishing it all up until it makes a distribution. Once they have that distribution, in this case the distribution is normal, they just use what we know about the shape of a normal curve. If a gene's FST value occurs in this tail, it's a higher FST than you would expect on average. If it is low enough, it occurs in this tail and it's lower than you would expect. And all of a sudden I was like, oh, I know how genomic people do statistical tests. They do statistical tests the same way I do them. But I had to realize that they're looking at FST as it varies across the genome from gene to gene. They squish that thing up to get to the distribution, and then they just run the same old statistical tests that I've always run. Except that they're doing it on tens of thousands of genes. Why did this figure appear in this paper? Because Patrick Phillips is one of the authors on this. This is how he thinks about statistics. When I was in graduate school, he taught me statistics. So I know how to think distributionally about things in the same way that he knows how to think about things in terms of distributions. And so as soon as I came across this paper, a light went off in my head and I'm like, okay, finally I get what these molecular geneticists are doing. I get what their rationale is for being able to test whether or not a particular part of the genome is more variable or less variable than you would expect. It all comes back to this. Whether you're trying to tell two means apart, whether you're trying to tell two regression lines to see if they have different slopes, if one regression line is different from another regression line, if you're trying to test whether a regression line has a slope that is different from 0, or whether you're trying to see if genes are more or less variable than you would expect. It's all the same thing. It's all the same idea. Which is why I told you the other day, what you're about to learn is the most important thing you can possibly learn in this class, because it equips you to learn, to understand anything that anybody ever throws at you. I was adrift at understanding these papers until I was able to put it into the framework of understanding how these things that they're measuring relate to a expected distribution. As soon as I figured that out, I was like, oh, okay, relax. So, questions? All right. We have a population. That population has an associated distribution of sample means. We don't ever get to know what this population looks like, generally speaking, because we don't usually have access to that population, because that population is too large to sample the entire population. So what we do is we take a limited sample from that population, And we know something about that population, not because the sample tells us something about that population, but because the sample has a sample mean, and that sample mean relates back to the distribution of sample means, and we know something from the central limit theorem about how that sample mean relates then back to the population. So we have to think about what is it about this sample that tells us something about the distribution of sample means. So when we take a sample, the first thing that we ever calculate on a sample is we calculate the sample mean. The sample mean is usually designated with the lowercase x with a bar over it. And the formula for this is that you sum up all of the individual x values and you divide by the number of values that you have. You've done this a gajillion times before. This is how you calculate your grade in a class, et cetera, et cetera. If you have a course that has 5 exams, each exam is worth 100 points. You score an 85, a 76, a 92, 84, 86, 1, 2, 3, 4, 5. To get your class average, if your class was based solely on exams, I can't remember the last time I taught a class where it was based solely on exams, you would just add all of these exams up. Whenever I use the uppercase sigma, this is an uppercase sigma, this basically just means sum. Add them all up, and then we divide by the number of exams that you have, and that gives you your grade for the class. That's the average. It's the arithmetic mean, it's the average, whatever you want to call it. That tells you where in this sample kind of the middle of the distribution of these sample points are. But that just tells you one thing. Essentially, when you do this, you are using the sample mean to hopefully tell you something about the population mean. But unfortunately, it doesn't really tell you much about the population mean. And the reason that it doesn't tell you much about the population mean is that this sample mean that you took could, just by random chance, have corresponded to the population mean. But depending on how whack-a-mole this distribution is, how much variance there is, whether it's bimodal or whether it's normal, that will affect the width of the distribution of sample means. Also, your sample size has an effect on the width of the distribution of sample means. Remember how when we were calculating the means, we were looking at samples and the distribution of sample means at different levels of sampling in that R script. As we got bigger and bigger samples, the width of the distribution of sample means got narrower and narrower the bigger our sample sizes were. So as sample size goes down, this distribution of sample means gets wider. And as we get higher and higher samples, this distribution gets narrower. Now what that means is that for large sample sizes, we kind of hone in on what the population mean is. For small sample sizes, we don't have as good an idea of where the population mean is based on any given sample. So that sample mean could have landed right here, but it could have just as easily landed way out here or way over here. You know, because we have a relatively low sample size and this distribution is broad. So the sample mean is almost never going to be the same as the population mean. The only way the sample mean will be the population mean is if you randomly stumble upon it. Because that's not what the sample mean is designed to tell you. The sample mean is designed to tell you, if you gathered that sample mean over and over and over again, you can make a distribution of it. The mean of that distribution then would be the population mean. But any given sample mean does not have the ability to tell you much about the population mean. It's a misconception that people teach all the time, that the sample mean is a person's attempt to estimate the population mean. If you think of it that way, there's nothing technically wrong with that, But just know that your estimate of the population mean from a sample mean is always going to be wrong. It will be somewhere in the ballpark, but where in the ballpark is really the question. Is it right there at the pitcher's mound, or is it somewhere way out in left field, your estimate of the population mean? So I prefer to not think of the sample mean as something that we calculate to gain insight about the population mean. I want to think of the sample mean as giving us some insight as to what the distribution of sample means is like. Because if I know something about the distribution of sample means, I can then look at another sample mean and figure out what I can know about its distribution of sample means, and then I can look at those two means and say, did those means both come out of a population that has the same distribution of sample means, or did they come out of two different populations, each of which has its own distribution of sample means? Is that clear? Okay. Hopefully I've indoctrinated you into kind of how I'm talking about things and thinking about things after this.
So this whole thing just works for population, right?
In the sense that all statistics is going to work with populations. So ultimately, like I said, ultimately it would be nice if we just had access to the entire population, but we don't. So we use sampling to give us insight into the population, but the insight that we get isn't about the population as much as it is about the distribution of sample means that comes from the population.
Mm-hmm.
So we can effectively forget about the population for a moment as long as we define what our population is. It doesn't matter what the shape of the distribution of that population is, as long as we can reasonably assume that this population will give us a normal distribution of sample means. And we can reasonably assume that because of the magic that is the central limit theorem.
Okay, so you said you can compare, you'll take a sample, you take a couple of samples, it builds this DSM.
Yeah, this is built off of tens of thousands of samples.
Okay. Each of the same size, I'll go.
Each of the same size, yep.
Okay. But then you said, rather than jump right from that up to the population, we can compare that to some other.
Yeah, so often,
What's the other, it's like some other
So-
date or some other experiment or something?
okay, so, I have give me a minute.
Yep.
Seldom do we give a rat's ass about the population mean. Like, who cares what the population mean is? Have you ever had a question where you wanted to know, is this mean equal to the population mean? I've been doing this since, okay, so when did I start graduate school? I started graduate school in 1989, so let's just say that I've been kind of a professional biologist since 1989. Before 1989, I took a couple of undergraduate statistics classes as an undergraduate. The only time that I've ever been asked to compare a sample mean to a population value was as an exercise in a statistics class. So that would have been like 1986 is the last time I've ever been asked to compare a sample mean to some hypothesized value of the population mean. Because we just don't do that generally. What we do instead is we do stuff like this. Just to give you some idea. gosh, there's some things that are not on my computer that normally used to be on my computer. I work on a project right now where I'm looking at an introduced species that has been introduced into the United States back in the early 1900s. It came to Texas in about 1950. In Texas, it was distributed along the Gulf Coast of Texas because it came in on ships. So it's a little lizard. It's a little Hemidactylus tursicus lizard. It's about this long. And over time, what's happened is it has spread. It's expanded its distribution, and it's moved north. Now it's found all the way up to Overland Park here in the Kansas City area. It's found in Pittsburgh, but traditionally it lives in the Mediterranean region of the Old World. So it came from the Mediterranean region on boats to the United States. Once it got to the United States, it expanded northward. And so I study a population down in Galveston, and I study a population in Edmond, Oklahoma, which is just north of Oklahoma City. The difference between these two populations is that the Oklahoma population gets very cold in the winter compared to Galveston, where it's actually quite nice throughout the year. And so one of my questions is this species evolving to cope with cold weather as it expands its range north? And so what I do is I go to Galveston, and I go to Oklahoma City periodically throughout the year. I go in the winter, and I also go in the summer, and I take temperature measurements of the environment that these geckos are in, and I also take their body temperature. I have a little gun that I can shoot at them, and it tells me what their body temperature is. And what we find, what I'm interested in is, are these two geckos drawn from the same overall population of geckos in terms of their thermal properties, the kinds of temperatures that they thermoregulate at, the kinds of temperatures that they're active at, or are they drawn from two different populations? I don't really want to know what the population mean is of the Galveston geckos and what the population mean is of the Oklahoma geckos. I just want to know if the Galveston population of geckos is different from the Oklahoma population of geckos. Well, if they're from two different populations, this will be maybe the best way of getting this across to you. If they're from two different populations, then the Galveston population will probably be warmer overall in terms of the environment that has available to itself, and the Oklahoma City population will be colder. So if we take samples from both of these, the sampling distribution, the distribution of the mean temperature, will be a distribution of sample means here and a distribution of sample means here. So when I go and take a single measurement of the body temperatures of geckos in Galveston and in Oklahoma City, those sample means will fall somewhere in this range for the Galveston, or for the Oklahoma City population, and they will fall somewhere in this range for the Galveston population. And all I really care about is, could I have gotten this sample mean from this underlying sampling distribution, and could I have gotten this mean from this underlying sampling distribution? I don't care what the population means are of those two things. What I want to know is, based on this sample mean and that sample mean, could these two sample means have come from the same underlying population or from two different populations? That is almost always the kind of question that we are asking in statistics. We're not asking, is this sample mean the same as the population mean? We're not asking if this sample mean is the same or different from this population mean. We're asking, is this sample mean different from that sample mean? One way of saying that is to say, is sample one different from sample two? Another better way of saying it is, could sample one have come from sample two's population? Could sample two have come from sample one's population? Because we're dealing with samples, we don't have to go back to the population. What do we go back to? a distribution of sample means that come from that population. We'll talk about how to do this on Monday. All right, any questions? All right, I'll see you on Monday then. I think I have everybody's coin flip data, so I'm gonna send that out to you as soon as I get it all put together, probably later on today. Work on that over the weekend and into the beginning of next week. Your coin flip assignment will be due on Wednesday. You've been unable to make progress on that because I haven't gotten the full data set back to you, because I haven't gotten people's coin flip data in a time.
Have we done just
Up to where we.
Yeah,
Yeah.
So we'll be working the rest of the